Hello everyone. Thank you for joining us today. My name is Jason Kilgore. I'm with Molecular Probes Tech Support. Today we're going to have an introductory presentation on the comparison of various basic immunolabeling strategies. A quick introduction. I've been with Molecular Probes, which is part of Life Technologies, for almost 13 years now. About 8.5 of those were with research and development, making dyes that were used for organelle staining and immunolabeling for microscopy. And the last 4.5 years or so, I've been with Molecular Probes Tech Support. We're joined today by Edward Lieber, who also helped with the deck. He's been with Molecular Probes Tech Support for 12 years now, and he'll be running the chat. So during the presentation, at any time, feel free to enter chat questions, and Ed will reply to those. And at the end of today, if there's still some time, we can stay a little later and continue answering a few more questions if there's time. If the time runs out, Edward and I will be answering some chat questions in the following day or two as well. Also, I wanted to tell you that at various slides during the presentation, there will be a survey going out to you. Feel free to answer some of the survey questions that come your way. Okay, let's begin. So, today we're going to talk about various immunolabeling techniques. The first presentation in this series was at the end of last year, where we talked about tips and tricks with immunolabeling and troubleshooting guides. And today we'll talk about the different strategies used for labeling. As you can see here, some of the topics that we'll cover. We'll talk about the differences between fluorescent and colorimetric labeling real quick, some basic guidelines for selecting an antibody, and then go into each of the different labeling strategies, their pros and cons. That's how they're what their problems may be and how they're beneficial. Some example images and comparisons between them. And then just a sort of simplified guide on choosing which of the strategies to use. First, let's talk for a moment about fluorescent versus colorimetric. When we say fluorescent dyes, we're talking about dyes that when you excite them with one wavelength, they absorb that energy and then emit a light at a higher wavelength. For this, you need a special fluorescent microscope with filters or lasers to excite the dye and filters to detect the dye. Colorimetric dyes refer to dyes that are a little more traditional and older. These are not fluorescent, but rather you see them in room light by the naked eye. You don't require special filters or cameras to detect them. These include like hematoxylin and eosin, hematoxylin being a dark blue color, eosin orange colored in room light for general cell labeling. Diaminobenzidine, or DAB, is an example of one which turns brown as an enzymatic detection. Colorimetric dyes have often been used with pathology, in part because of the stability of the signal. Once you've done the labeling with the colorimetric dye, it's very stable. It doesn't photo bleach away. And on the tissues that you're labeling, the signal is retained very long term, so it's archivable. Fluorescent dyes are not as stable. They will photo bleach when you shine light on them, although you can mediate this to a large extent using an anti-fade mounting medium. And there's a number of commercial anti-fades available. Ours are Prolonged Gold, which hardens, and Slow Fade Gold, which stays liquidy. But even with those, sometimes the colorimetric dyes are more stable. However, one of the benefits of using fluorescent dyes is the signal to noise. Because you have a black background and you're able to select out only a particular wavelength, you can get a much better signal to background, which improves your sensitivity. With colorimetric dyes, sometimes sensitivity is an issue. Also, for multiplexing, because fluorescent dyes are much brighter, and you can select out only particular channels, particular dyes at a time, you can now do more dyes at a time on a single sample. With colorimetric detection, usually you're only talking about 1 or 2 colors, sometimes a third. With fluorescent dyes, you can get up to 4 or 5 or sometimes even more with some special techniques. Cost-wise, fluorescent dyes do tend to cost more, given the amount that you get for a certain price. However, with those benefits for sensitivity and multiplexing, it's often worth it. Okay, so in terms of labeling strategies that you can use, I'm not going to talk about all of the ones that are possible, but these are the most common. So what I'll do, I'll talk about each one separately, its benefits and possible challenges, a definition of it, and then an example image using a common antibody and the protocol. And so I'll be comparing all of these qualities with each of them. First, we'll talk about direct conjugates, and then the standard secondary labeling that's most commonly used, at least for microscopy. And then we'll go into a couple of amplification techniques, Avidin-Biotin, as well as enzymatic amplification like pyramide signal amplification, or TSA. And then just briefly mention some of the combinations that you can use. Obviously, the star of your protocol is your primary antibody. This is what everything hinges on, right? And so you have to select your primary antibody very carefully. First off, you want to make sure that, not only is it against the target that you're trying to detect, but pay close attention to the primary's species that it's been validated for, right? You have your host species, and then you have the target that it was raised against. Also, there are a lot of different commercial sources. We have an antibody selection tool for primary antibodies on our website at lifetechnologies.com. There's also some other general websites that you can go to, like linscottsdirectory.com, as well as biocompare.com. In choosing your primary, of course, you also want to consider what sort of instrument platform or samples that it's been validated for. For instance, has it been validated for microscopy or immunocytochemistry, or only for Western blotting? It makes a big difference. They should also have been tested for immunofluorescence. So if you're doing a fluorescent labeling, check to make sure that immunofluorescence has been used with it. And of course, pay attention to the protocols that are recommended for that antibody. For instance, the fixation type can matter a great deal. Some antibodies have been validated only for formaldehyde fixation. If you do a methanol fixation, for instance, the resulting delipidization of the sample may remove the protein or the antigen of interest. Other antibodies prefer a methanol fixation, for instance. So you need to check that. Some antibodies also require an antigen retrieval technique to unmask the antigen after fixation. So there's a lot of different concerns, and I covered those mainly in the first webinar in this series at the end of last year, which you can still find online and can register for and view online at the same site where you registered for this one. Okay, continuing on. Let's go on into the different labeling strategies now for how you can detect your antigen. By the way, you will be getting the first of your surveys There will be 5 different surveys going out during the procedure of this webinar, and the first one should be pushed to you now. Okay, so direct conjugates of primary antibodies. When you hear someone talk about a direct conjugate, what that means is that there is a primary antibody which has had a reactive dye conjugated to it covalently. So there's no secondary detection. It just already has the dye on it. This allows some certain benefits. First off, it takes much less time to do the labeling because now you've done the fixation and permeabilization. You do your label step with your conjugated primary, and then you can mount and view. So it's much quicker. The label time can only be maybe an hour or so, including with washes. Also, if you have multiple primaries that you're trying to use which are of the same species and same isotype, now you don't have to worry about some potential problems with cross-reactivity. So for instance, if you have 2 mouse primaries that you're wanting to use, you can directly conjugate them or purchase them already conjugated and use them together in the same labeling step. The problems with these, one is that they're only about half the intensity of a standard secondary labeling. So if you're trying to detect a low-expressing antigen, you may not be able to get a very reasonable signal over the background. This is especially a problem with tissue, because tissue often has autofluorescence problems that decrease your signal to background already. In more sensitive detection methods, such as flow cytometry, because that platform is more sensitive, direct conjugates are often used. With microscopy, it's less common to have a direct conjugate because it's not as sensitive and sometimes there's autofluorescence issues to overcome. Also, because you've now conjugated dyes onto the surface of the antibody, the size of the complex is a little bit larger. This can lead sometimes to penetrance problems into your cells, especially into the nuclear compartment or into your tissues. And this means that you need to permeabilize better and have longer label times to overcome this. Now, I mentioned that some antibodies come already conjugated with a dye in the marketplace, but we also have antibody labeling kits that you can purchase and use to label your antibody with a dye if it's not available commercially that way. We're hoping to have a webinar on that sometime later this year. This next slide shows an example, and in each of the different labeling strategies, you'll see a picture like this one of BPAEC labeled cells, bovine pulmonary arterial cells, which are labeled with an anti-tubulin antibody. So what you're seeing here are cells that are labeled for tubulin. It's a very common antibody and a fairly high expressing target. Here is an example where I've labeled the tubulin with a directly labeled antibody. So the anti-tubulin is labeled with Alexaflor 488. And then we incubate the cells with the protocol on the right. So you fix and permeabilize, wash, and then block. This was actually blocked with 6% bovine serum albumin and a 5% normal goat serum, or NGS. And then there's an incubation where the primary is in the blocking solution. Only takes about 30 minutes for cultured cells. Then you wash and then mount with a cover slip. So the protocol there doesn't show the washing steps. The entire time takes maybe an hour to an hour and a half, so it's fairly quick. But you can see here this used a 60x objective, and it still took 500 milliseconds, which is relatively long. So it was not the brightest of signals, and that's one of the drawbacks. I'm actually going to skip ahead a slide So that was primary antibodies. For secondary antibodies, the secondary antibodies labeling, this is where you have your primary antibody, which does not have a dye on it. So you label your cells with your primary antibody, which is unconjugated. And then you come in with a secondary antibody that has a dye on it. So for instance, if you have tubulin as your antigen, you come in with a mouse anti-tubulin antibody, primary, and then you label that with a, for instance, goat anti-mouse, secondary, that has a dye on it. This is the most commonly used technique, arguably, at least for microscopy, for labeling antigens, in part because so many primaries are not available as direct conjugates, but also because this gives a brighter signal than a direct labeled primary. There's about 10 to 20 dyes per target once you've done the secondary labeling. And there's a large selection of commercially available conjugates for secondaries. Again, on lifetechnologies.com, there's an antibody selection tool for secondary antibodies that we have. And there's a lot of different species, so whether you have a rabbit primary or mouse or human, there will be usually a secondary antibody to match that in whichever color that you want to choose. The problems compared to a direct conjugate is it takes a little bit longer because now you have 2 steps instead of 1 for the labeling. You have to check the potential for cross-reactivity of the species if you're doing multiplex labeling. So if you have a mouse primary and a rabbit primary on the same sample, you have to make sure that your secondary antibody that you're using to detect them with doesn't label the wrong species. So you don't want your goat anti-mouse to label your rabbit primary or your goat anti-rabbit to label your mouse primary, right? So you have to check when you purchase your secondaries, you have to check and make sure that they've been cross-absorbed against the other species to minimize that cross-reactivity. Sometimes when secondaries come to you, the manual will say what they've been cross-absorbed against. If not, it's a good idea to call the technical support and ask about it. Also, with every additional step, you have the potential for additional nonspecific background labeling. When you have a primary antibody and you label with it, sometimes there will be some background labeling. You want to block with BSA and normal goat serum, for instance, to help minimize that. But you also now, because you're doing a secondary antibody, there is the chance that the secondary also can lead to increased background. So you have to block that as well, such as with 6% BSA. I'm going to go back 1 slide here and talk about choosing the secondary antibody. Now, how do you choose a secondary antibody? Well, there's a number of different types for certain usages. The most common is a whole IgG. That's at the top of the list there. This gives the strongest signal. It's the least expensive. There is some possibility for cross-reactivity, but most of the secondary antibodies that you'll use are of a whole IgG type, and those are conjugated with the dye, right? Also, sometimes there are Fab2 fragments, which won't interact with a Fc receptor. It's not often that you need that for a particular labeling, but it's there if you do. It can give a little bit lower of a signal because it has fewer dye molecules on it. Sometimes the penetrance is a little bit better with them because they're smaller. If you're doing multiplex labeling, that is, if you have multiple primary antibodies of different species, often it's a good idea to use a highly cross-absorbed version of the antibody. That is, it's been cross-absorbed against more species to limit the cross-reactivity. It may require a slightly higher concentration when you use it, and it would probably be a little more expensive than the whole IgG option. But if you're doing multiplex labeling, it's the way to go. And finally, there are some secondary antibodies which are subtype specific. So, for instance, if you're doing two mouse primaries on the same sample, if one of the mouse primaries is an IgG1 isotype and another is an IgG2A isotype, then you can use subtype-specific antibodies to label them in the same step. If you don't, if you just use whole IgGs, they will cross-label and you'll get both antigens labeling with both colors, and you definitely don't want to do that. Okay, can everyone hear me okay? I'm getting a comment here that I'm not being heard well. Can everyone hear me? Please enter a chat comment. Okay, well, I'm going to continue on then. Okay, so those are some of the considerations for choosing a secondary antibody. And the same sites that I mentioned before you can go to help find a secondary. Again, we have a selection guide at lifetechnologies.com, or you can go also to biocompare.com or linscottsdirectory.com. Okay, continuing on. You'll also potentially be getting a survey here at this slide. Okay, here's an example of some cells that have been labeled with a secondary antibody. Again, this is tubulin, as with before. If you remember, the directly labeled antibody had an exposure time of 500 milliseconds. Here we have the same objective, but now it's only 40 milliseconds. So you can see it's much brighter than the directly labeled. On the right, you see a typical labeling protocol. So you have your fix and perm. You block, label with your primary, maybe 30 minutes, wash, and then label with your secondary antibody for 30 minutes, wash, and then cover slip. Again, tubulin is a highly expressing antigen, so it gives a very nice signal here. Okay, on to Avidin-Biotin amplification. So, if you're trying to detect a low-expressing antigen, that is, there's not many of that protein present in your sample, doing a standard secondary detection may not have been able to detect it very well above background, in which case you need to go to an amplification method. Probably the most common way of amplifying your signal is to use an Avidin-Biotin or a strep Avidin-Biotin labeling. This is a 2- or 3-step protocol where either your primary antibody or your secondary antibody is labeled with biotin, which is a haptin. Then you come in with a strep Avidin or an Avidin that is dye-labeled, and that detects the biotin that's on it. The end result is that you get about 20 to 40 dyes on your target, so now you have a much brighter signal than you would with a standard secondary detection. This is a fairly traditional method. It's been used quite a lot and published on very commonly. It's much brighter, and low abundance targets are much more likely to be detected. However, it is a longer protocol, so now you can have up to 3 different steps, each one taking maybe 20 to 30 minutes, plus washes and fix and perm. And as I mentioned before, with every additional step, now you have the potential for nonspecific binding increasing. So you need to block each step with BSA. Also, cells have endogenous biotin, mainly in the mitochondria. So if you have cells and you do a control with only the strep Avidin, you're probably going to get some biotin labeling in the mitochondria as well as your specific labeling. This can be a problem, but there are blocking solutions, and we sell a couple of them, where you can block the endogenous biotin prior to doing your primary antibody. Here's another example image, again of tubulin. this time labeled with a biotinylated secondary, a biotin anti-mouse, a goat anti-mouse, and then detected with streptavidin alexaflor 488. This had an exposure time of only 20 milliseconds, so now this is twice as bright about compared to the standard secondary detection where it was about 40 milliseconds, right? Compare that to the 500 milliseconds needed for the primary directly conjugated, right? On the right, you can see the protocol that was used and how it's a bit longer now, so it takes a little more work to do. And you'll also notice step 2, where it has endogenous biotin blocking. And that step, it's a 2-step process that takes about 40 minutes or so. So we've added a fair amount of time there. But we have a much brighter signal. Now, tubulin is already fairly high expressing, so you don't normally need a streptavidin biotin labeling for it. But for many low expressing targets, you will. And I'll show an example later. Okay, and I think you're also getting a survey now on this slide. All right. Now, the final method that I'm going to discuss is enzymatic amplification. Enzyme amplification means that you have a primary or secondary antibody that has an enzyme conjugated to it, like horseradish peroxidase, for instance, or alkaline phosphatase. You then come in with a substrate to detect that enzyme, and that substrate will then deposit around the site of your antigen, giving you a much brighter signal. For traditional colorimetric dyes, alkaline phosphatase is often detected with, for instance, BCIP-NBT, which gives you a blue room light color, right, on your sample. If you have horseradish peroxidase, that's often detected colorimetrically by DAB, or diaminobenzidine, which gives you a brown color. But for fluorescence, horseradish peroxidase is the most common, and one very good way of detecting HRP is to use a tyramide signal amplification, which I'll talk about in just a moment. And this will using a TSA amplification, you'll get about 120 to 200 dyes per target. So that's a very bright signal. This is the brightest option for labeling with an amplification technique in fluorescence. Very useful for the most low abundant targets. And this has even been used for chromosome detection, targets on chromosomes. It can bring out fine structure in tissue sections or chromosomes. The problems are, you have more steps now, so you have an increased protocol time compared to the previous methods. And with more steps comes a greater chance of nonspecific signal as well. With an HRP detection, now you have to be concerned about endogenous peroxidases in your sample, especially with tissue. And you can block that. So before you do your primary antibody and blocking, you do a separate blocking step for endogenous peroxidases with hydrogen peroxide, like a 3% hydrogen peroxide wash, which will kill the endogenous peroxidase. And then you go in and label with your primary and your HRP secondary and detect that with your TSA. Here's an example on cells. Notice the exposure time now is only 10 milliseconds, so very bright. You see on the right-hand side a much longer protocol now. You have your primary, your secondary, and your TSA amplification. Sometimes you can combine methods which will make it even longer. One drawback on cells, on cultured cells, is that sometimes your target, now you'll start to lose a little bit of resolution with TSA. This is because it's not labeling the antigen, it's actually depositing the dye around the site of the antigen, which can lead to a little more of a slightly fuzzier image, a slight loss of resolution on cells. This is not true on tissue, interestingly, as you'll see in the next slide. Also, you may be getting a survey now, by the way. Here on this slide, you can see that on the left-hand side, I've labeled this cerebellum mouse cryosection with a tubulin antibody, which is labeling Bergman glia. A very nice resolved structure there, and you can see some astrocytes labeling in the middle of that cerebellum image. This is very resolved, and one reason may be because of the complexity of tissue. Everything is much more compact, and so you're able to get a more resolved image. On the right-hand side, you can compare this to a standard secondary detection with a goat anti-mouse, Alexa 488. You can see much lower signal to background compared to the background signal there. And the exposure time, 200 milliseconds versus only 40 with PSA, so you get a much brighter signal. Here's just a graphic that compares all of these different labeling strategies. You can see as you move from on the far left-hand side, you have a direct conjugate. Then in B, you have a standard secondary detection. You've gone from 2 to 8 dyes in the target for the direct conjugate, now to 10 to 20 for a secondary. With a biotinylated, now you have 20 to 40 dyes per target. And finally, with a TSA detection, you have up to 200 dyes per target. As you increase to the right, you're increasing the processing time and potential background issues, but you're also increasing your sensitivity. Let's look at some other images now. These were generated by a colleague in our research and development group, looking at high expressing targets versus low expressing targets with 3 of these different methods. At the top, again, this is tubulin, like the other images I had shown you, which is a very high expressing antigen. A direct labeled primary antibody on the upper left hand works just fine with this for labeling cells in microscopy. It's a little bit longer of an exposure time, but you're able to resolve the tubulin fairly well. More commonly, in the center top there, you can see a goat anti-mouse standard secondary detection. That works very well. It gives a reasonably bright signal, bright enough for any common imaging. And then the upper right-hand corner, now you see TSA labeling. It's super bright, 5 milliseconds, even with a 10% neutral density filter thrown in. So it's actually too bright for standard imaging, and you've lost some resolution. However, if you go to a low expressing target, like on the bottom there, in this case alpha-catenin, which is a marker for cell-cell junction labeling, you'll notice that a direct labeled primary on the lower left-hand side doesn't work to detect the antigen above background. You can't even see it in the cell-cell junctions, and you start to see more of the autofluorescence and background at that really long exposure time of 1,250 milliseconds. So that's quite a long exposure. Using a standard secondary antibody in the middle there, you start seeing your target. You can see some striated lines between the cells there. That's your specific target, where you have your cell-cell junction. The blue here is DAPI, by the way, for nuclear labeling. But to get a really high-resolved image of the alpha-catenin, now we've used tyramide signal amplification on the lower right, and now you're able to get a very specific, good signal to background with only a 15 millisecond exposure time. So in the case of alpha-catenin, an amplification technique such as TSA or maybe Avidin-Biotin detection is the way that you would want to go. Whereas with a high-expressing target like tubulin, a standard secondary is probably most commonly used. Okay, so the next slide. So those are the 4 main labeling strategies that I wanted to talk about today. But how do you go about choosing them? Here's sort of a simplified decision tree that you could use. The main question is, how abundant is your target antigen? Is it very abundant, like tubulin, or very low abundance, like alpha-catenin, for instance? Are the cells that you're detecting very rare, and is the target very low expressing? If it's a low expressing target, then you're going to want to use streptavidin, or maybe even tyramide signal amplification there on the left. Most targets will use a standard secondary detection, so sort of a mid-level there. And if you're doing imaging, microscopy, right, you'll choose a standard secondary. If it's a very high expressing target, you can go on and potentially use a direct conjugate. If you're doing flow cytometry, which is much more sensitive, you have more choices, right? You often direct labeled primary is fine, although secondary detection works very well too. If you're doing multiplexing, that is, if you have multiple primaries that you're detecting on the same cells, you have to give some thought into how abundant those targets are, because most likely you're going to want to do some sort of direct conjugates, where you label one of your primaries with a green dye, for instance, and then the other with a red dye, and label them together on the same sample. But if your sample, if your antigens are low expressing, that's not going to be an option, right? Because that direct conjugate, at least for microscopy, is not going to work so well above background. Now, the 4 strategies I told you about, you can do combinations of these to get an even greater amplification method. For instance, you could do a primary antibody, come in with a secondary that's biotin labeling, then detect the biotin with a streptavidin that has HRP on it, or horseradish peroxidase. Now you've done an Avidin-Biotin labeling. And then you detect the HRP with a tyramide signal amplification method. So now you've got a 4-step protocol with a double amplification, if you will, to get the brightest possible signal. Of course, now you have to deal with endogenous biotin, and you have to block with that. You have to deal with endogenous peroxidases, so you have to block for that too. There's a longer protocol and a potential for increased background, right? So these are risks that you have to consider if you're trying to do the amplification and combining some of these strategies. Okay, so just a real quick summary. You have to tailor your labeling techniques to the conditions that you're doing, right? Most notably, how abundant is your target? Do you have to do multiplexing, and will there be cross-reactivity between your secondaries? And considering the backgrounds that you have, that in the longer protocols compared to the need to amplify your target, right? All right. So I hope that this presentation has been useful for you in introducing some of these labeling techniques and some of the pros and cons of these. You should be getting also a survey, probably a couple slides ago. But we always love to hear feedback from you. We're going to continue to monitor chat here for a few minutes and try to answer some of these questions. But if we're not able to get to your chat question in the live presentation, feel free to send us an email or call us here at Molecular Probes Tech Support. And if you're one of our European customers also, you can send to the same tech support at lifetech.com, and it will come here to us. My name is Jason, but anyone here can help you. There's also a phone number which isn't indicated here for our European customers, but you can find it online. And if we don't get to your chat questions today, we'll try to follow up with email with some of them in the next day or two. Okay? Thank you very much for attending this webinar, and we look forward to having you as continued customers. Bye-bye now.